that I might post this for fun, talking about sleep, that they actually look at dendritic spines, you wake versus sleep, and they see them changing based upon your level of consciousness. And so this is really a lot more plastic than you might think as far as these dendritic spines. But for us, it's the idea, it increases surface area and allows, so there's plasticity then, more input. And that's what they do. And what the dendrites are going to then do, they're going to send this information to the next part of the neuron, which is the cell body. Now the cell body is sort of the traditional cell we draw. When we did the cell chapter, we said there's a nucleus, endoplasmic reticulum, all that stuff. That's actually where for the cell, the bulk of that information is. It's the cell body. So it has that bullet point. The other bullet point that we're going to see today is all this information coming in. We're going to have excitatory information, like let's go, and inhibitory, because we don't want to be off jerking around. So the idea is integrating these, and we'll see this, the cell body adds up these signals and sends them to the axon. And then we're going to see at the axon, we're going to have an all or nothing thing. It's either going to fire or it's not. And so, again, that's sort of, we'll look at the details of it, but that's sort of what the cell body is going to be. It's going to make everything, it has a nucleus, but it's also taking in all this information on the way to the axon. That's what integrates. Finally, the axons are the extensions, okay? So these, again, can be very, very small if you're going from, an interneuron in the brain to the other, or it could be a huge one if it's going from your brain controlling my toe to move. So there's various lengths there. And that's in 3 parts. Again, starting off sort of simple. The picture here is not to memorize because we're not in anatomy, but we're going to keep talking kind of about a neuron that looks like this. Okay, and we'll talk about myelination in a minute. But, you have the dendrites, a nice cell body, and an axon, and we'll get to the end here. But if you look at different nerves, they don't really all look like that. And so they have very different structures to match their function. If you look at this one, this probably represents a neuron that's in your cerebellum, or some part of your basic brain. And so it's receiving hundreds of thousands of inputs to actually control, basically, coordination and that I don't fall down. So if you look at this dendrite, it's a huge dendritic tree. Okay, so very specialized. You can see some of these, it's actually hard to see which ones are the dendrites and which ones are the axons. Sort of, like that. Sensory ones, the cell body's in the middle. So you're not memorizing any of these. I just want you to realize that, this isn't what all, neurons look like. But we're going to keep drawing. Well, mine looks kind of weird anyway. But we're going to keep drawing and talking about that. Okay, the next part, and we're going to make it to almost the end today of this, because we're going to talk about how electricity travels through here. But when we talk about the nervous system, we're going to be talking about it being electrochemical signaling. Because what we see here, and almost all of our neural connections are, we reach to the end, so we reach to the nerve terminal, but the cell that it's communicating to is actually not connected. There's a space there. And this space, and we've all sort of heard this term no matter what, synapse. The synapse. And so if you look at it, and again, I do have we sort of blow it up here, you're going to have the presynaptic cell where it ends, the postsynaptic cell, this time it's another neuron, it could be a muscle or a gland, and you have that synaptic space in between. So somehow, we've got to get a message from this cell to this cell, and there's a space. So although we're going to talk about electricity flying through here, there's actually chemicals here that are going to be released called neurotransmitters. And they're basically going to bind to receptors, bind to proteins in the postsynaptic cell, and we're going to have some sort of response. So it's electrical, but it's chemicals, but there's neurotransmitters. What are some neurotransmitters, you think? Say again? Well, GABA, of course, because that's a real cool one that we're going to talk about. But what's ones we might have heard of just on TV watching more, like the little dog that's sad, serotonin, dopamine, which we're going to talk a lot about drugs. We're going to talk about those, norepinephrine. Those are some typical neurotransmitters. And really, that's why I'm sort of, the drug video we're going to do is actually a little ahead of the game, but we'll catch up to it. When it comes down to it, those type of neurotransmitters control a lot of our, a lot of activity, but also control a lot of our moods, anxiety, etc. So, imbalances of those seem to be related to bipolar and depression and that type of thing. So most drugs work sort of to modify sort of those neurotransmitter levels, and we'll actually see that. These include prescription drugs and illegal drugs. And you'll see, really, there's a lot, the mechanisms of them are very similar, and what's illegal, what's prescription is sort of arbitrary. I'm not telling you to go out and use drugs, but you're going to see when we talk about, anybody know what SSRIs stand for? And we'll see this selective serotonin. I just had to give a drug preview here. Don't worry about this yet. Reuptake inhibitors. So we're going to see sort of control of serotonin involves putting it back, reuptake. And the trouble is a lot of disorders, we don't have enough serotonin. So we want more serotonin. So we inhibit the reuptake, and boom, we have it. That's like Prozac. We're going to see ecstasy actually increases serotonin levels in a similar fashion. Obviously, the effects of ecstasy are are greater, I guess. I've never done ecstasy. But that's, I'm like old. I do old drugs from the 70s, okay? This ecstasy stuff, it's like, I don't know what this stuff is, okay? But it actually works a lot like Prozac, at least the mechanism, and that's sort of like weird, okay? So again, don't, if your parents are on ecstasy, don't say, or I'm sorry, if your parents are on SSRIs like Prozac, don't say that you're on ecstasy. My teacher said you're like on ecstasy, Mom. No, I don't need that. Okay, all right. So control, this is important, and we're going to see that in the drug video and later. This is sort of a picture, and you get the idea now. The presynaptic cell and the postsynaptic cell does not look the same, because you have all these vesicles with all those neurotransmitters that when the electricity hits there, and Monday we'll get to the mechanism, that's going to release the neurotransmitter, bind to this next cell, cause the cell to fire or the muscle to contract. Okay? And so this is sort of the whole idea. And really, this chapter that we're going to start today and finish on Monday literally will take us through a signal all the way to what happens in the postsynaptic cell. And then the drug video also will sort of, carry with that. That's what's cool about this chapter. That's mainly what this chapter is, from beginning to end. So, of course, we're revisiting this. But before we do, I promised you that we were going to just mention each glia and one thing to remember. And then we can get to the fun stuff. And believe me, it's going to be fun. So, we're not memorizing tables, of course, or anything like that. But I'm going to be talking about 4 types of glia and what they do. First one. This is the only glia I'm going to talk about in the PNS, are the Schwann cells. We're not talking satellite, Schwann cells. And so Schwann cells myelinate peripheral nervous system nerves, PNS nerves. We're going to see this later today as well. If you look at myelination, now right now it's Schwann cells because it's peripheral nervous system. You actually see it's like wrapping membranes. When we know what membranes are. Can a lot of things get through membranes?
No.
No. That's going to help insulate these nerves because we're going to see nerve firing are just ions crossing membranes. And of course ions don't normally do it unless we have sort of channels to open. So you're going to see if we'll look at the function. When we look at Schwann cells, they're myelinating these regions, or the axons of peripheral nervous system, PNS neurons. And that's going to speed them up, we're going to see. They're quite a bit faster. So that's PNS. Do you think there's any myelinated nerves in the central nervous system? What do you think? Yes. And the oligodendrocytes do that. So the oligodendrocytes, the bullet point is that they myelinate CNS neurons.
Is the second type of
And that's the second type of glia. So we're going to have one PNS one, which we've done, And now I'm going to do 3 of the CNS, this being the first CNS one. Okay, so oligodendrocytes. Sorry, I didn't mean to make noise with ice, I just didn't melt it. Oligodendrocytes are myelinating CNS neurons. Now, astrocytes, they do a lot, as you can kind of see. They're actually very important. You know, these glial cells are really important for nervous system function, but again, we're not going to talk too much about them. The only one I want us to remember, and I have a couple of pictures, are that these astrocytes are really important in forming the blood-brain barrier. When you look at most vessels, and we talked about them in the last unit being exchange, you know, in most places in the body, there's easy exchange between the blood and the tissue. There's sort of spaces in between the cells that enable things to go. If you look at the blood-brain barrier, which is a more restrictive type of exchange, you actually see the endothelial cells themselves. The cells that make up the blood vessel are connected by tight junctions. So already it's hard to get things from the blood, across into the brain. But not only that, but these astrocytes basically form a whole glial layer. And so that things have to not only get through these endothelial cells and the tight junctions, they've got to get through a whole layer, you kind of see it all wrapped down, a whole layer of glia, these astrocytes, to actually make it into the brain. So I thought that was sort of cool pictures. You can see it here too, making it through both of these barriers to actually get into the brain. And that's what I want us to remember about astrocytes, blood-brain barrier. Now, the last one, which is the 4th one total, the 3rd CNS. If you look at the situation of, that it's really hard for things to get into the brain, if you talk about the immune system, which we kind of will a little bit later, blood cells, circulating white blood cells, have to actually make it into the tissues if you have an infection. And they're able to get through those blood vessels. It's hard to get through the blood-brain barrier. So we have a glia which serves as sort of the immune function, the immune cells in the CNS that are already there. These are microglia. And they're going to be scavenging, and and remember that cell that was crawling around, and the bacteria. They're going to be doing those jobs in the CNS because of the blood-brain barrier. So microglia, and that's it.
Wait, what are they? They're going to be crawling around.
Well, they're going to crawl. that if you have any sort of infection, or if you have some dead cell, or any kind of invader, just like most white blood cells do. They're going to be going to it and engulfing it, like what happens, the macrophages, etc., in the rest of the system. Those 4 types. Okay. Now comes the fun. Because you're asking, okay, well that's fun, Michael. What causes electrical signal? And what causes it? And this is going to be. Part of this is going to end up being what students have called the circle of life, and we'll see what that means in a minute, is stuff we've already talked about. Okay, these are bullet points, but we're going to sort of make it kind of come to life here. The idea is, like all cells, cell membranes, they're semi-permeable. Basically, some things can get in and some can't. And really, the main things we're talking about that can't just flow across without some help are ions and proteins. Now, if they were equal on both sides, if we had an equal amount of sodium, and let's say an equal amount of potassium on both sides, would that really be any potential to do anything? That's be like we have a dam up there, but the water's all down here. So we want to create, and we'll see this, there's an unequal distribution of these. And this is going to be potential energy. So we're going to do this in two layers here. This is a cell, almost all cells, but these are nerve cells. What the key thing inside is are going to be negatively charged proteins. In fact, I kind of want to put that picture, not that picture, this picture. Because this is going to be how nerves and muscles work, right here. We're going to talk about 4 ions and a difference in charge between the inside and the outside. And this is one, and, we're gonna concentrate on these two in a minute. But this is basically what some students call the circle of life. If you talk to Lynn, because once you draw this on your page or start doing it, you're going to be able to answer a lot of questions about how an action potential works, etc., without memorizing it separately. And so, overall, when we're looking at these cells, there's these 4 ions in this. There are negative charge proteins inside the cell. Okay, negative charge. Can proteins just pass out? No. They're going to stay there. Okay? Now we're going to show you how we create this, but basically there's a high concentration of potassium ion inside the cell compared to out. Okay, we're not even going to put that there. And then there's 3 ions which have a higher concentration out versus in. And those are going to be sodium and calcium and chloride. And overall, for a typical neuron, which is what we're going to be kind of working with, if these are the ions, but if you add up the charges of everything, the inside of the cell is what compared to the outside? Negative. And we'll show you kind of the way to measure it. It's basically negative 70 millivolts. There is potential here. There's 2 types of potential. There is basically chemical potential, because, okay, sodium cannot cross, but if we open a sodium channel, which way is it going to go? Down its concentration gradient. If we open a potassium channel, which way is it going to go? Out. Okay? And when you look at sodium, not only is it going down its concentration gradient, but what do we have here? Electrical gradient. Positives attracted to negative. So because we've created this, and I'm going to show you the 2 things you need to know of how we created this, because we've created this, putting the water up at the dam, basically we've got a ton of potential energy. Electrochemical gradient. Okay? Chemical, here's the electrical, plus minus. So that when we open a sodium channel, sodium's going to rush in. We open a potassium channel, potassium's going to rush out. That's going to be how a nerve fires, those 2 ions. Literally, sodium rushing in, potassium rushing out, using channels. That's literally how an action potential will travel all the way through the body. Just those 2 ions, just those opening up that channel. So this is going to be the key. That we're going to come back to this when we start just asking questions. But that's what you have to know, just like down in your brain. That if you compare the inside of the neuron to the out, it has a lot of negative charged proteins. Potassium is high, and then these 3 ions are high outside.
Higher, right?
Higher, but high. Yeah, I mean there is some ions there, but it's a much lower concentration. That's potential energy. Again, electrochemical gradient. The chemical based upon the concentration of these ions. Electrical because of this positive negative. So this is a nice little thing we created. We created a big, basically, potential energy to actually use. But was this just natural? Did this just happen? Like, oh, we got lucky? No. We've got to work at doing this. We've got to create, basically, before we fire, we have to create something called the resting membrane potential. The resting membrane potential of the neuron, of the cell. This means resting in the sense that we're going to do some things to create sort of an equilibrium here, that it's basically ready to go. And when it's ready, when we signal it, when we trigger it, it's going to fire. But then when we're not triggering it right now, it's setting the waters in the dam waiting. We're at an equilibrium. This is called the resting membrane potential. And there are going to be 2 things. This is actually, when we do this, actually let me just kind of show kind of when we're putting -70 how they did it. It's not that important, but the idea is if you put a probe inside and out, the inside is more negative than the out, and again, it's about -70 millivolts that you're measuring. So how did this happen? How do we create the resting membrane potential? Well, first, if you're reading the book, what I'm about to describe is actually at the end of chapter 5. Because we were talking about membranes in chapter 5, and we didn't even do chapter 5. Luckily, I didn't do it then anyway. I always do it here right at the beginning of the nervous system. So, no foul. And really, those are the 2 I want. So to examine this, let's get rid of the calcium chloride. Because those are not really the ions involved in making the resting membrane potential. It's sodium, potassium. And we're going to list two things. We're going to simplify this whole thing of what causes the resting membrane potential. One. Okay, well, how do you think we get all that sodium out and all that potassium in? It's going to be something called the sodium-potassium pump. Okay, it's going to use ATP, it's going to use energy. And so this is a pump that's in every one of our cells working 24/7 to do something. And what it's doing is, it's pumping 3 sodium out and 2 potassium in as it works. So as it's doing this, it's going against the concentration gradient, so we're getting rid of all the sodium, a lot of it obviously, but we're doing it where we're pumping 3 sodium out for every 2 of potassium we're putting in. And we're creating these 2 huge gradients, sodium, potassium, waiting to do something, right? We put the water in the dam, that's potential energy. But let's look at charge, because we also care about the inside versus out. If we're putting in, or if we're taking out 3 positive and replacing it with 2 positive, what is the inside of the cell going to become? Negative, right? That makes sense. That's the first, number one of how we're starting to create this electrical gradient. Okay? The sodium-potassium pump pumps 3 sodium out, 2 potassium in, keeps doing that till we're at a certain level, and that's making the cell more negative. That's sort of easy. It's 3, 2. The second one we've got to think about, we've got to look at more closely. Okay, so that's number one. The second one, again, you can kind of see the sodium-potassium pump working. We're pumping sodium against its concentration gradient to get it out here, potassium against its concentration gradient to have it in here. Now the second one has to do with the following. There are channels called leak channels, and we're going to actually see all these channels. So leak channels, and there'll be, again, it's even on your handout, it'll be in a few slides. Leak channels basically allow the ions to leak back down the concentration gradient. Okay, we're not going to trigger it really, they're just going to be able to leak through. So all we're caring about is sodium potassium. Now, if you look at this, let's just kind of basically see what would happen. Okay, we open up a leak channel for sodium. Which way is sodium going to flow? Back into the cell, right? We created this gradient, all the sodium out here. Sodium, if we allow it to leak in, it's like putting water in the dam and having a leak, right? It's going to leak through the hole and go down. So, with the leak channel, sodium will leak in. Okay, does everybody see that? Because that's going to be important for everything. That's where you use the circle of life, the idea that if we open a channel, you have to know 2 things. Which way does it go, and what does it do to the charge of the cell? Which way does it go, and what does it do to the charge? So, we open a sodium channel, or we don't open it actually, this time there is a leak channel. Sodium is going to leak back in. And as it does that, what is it going to do to inside of the cell? Make it more negative or more positive?
Positive.
Okay, so the inside will be more positive when this happens. Okay. Let's just look at that. Okay, now let's look at the other one. If we open a potassium channel, or if we allow potassium, to leak, it's going to leak out. Okay? Potassium leaks out down its concentration gradient, so potassium will go out. And then the cell will become, the inside of the cell,
Negative.
more negative. Okay, but let's look at this closer. I'm going to have a figure, but let's just look at this right now. Let's look at sodium again. Now, if we allow sodium to go, it's not only going down right now its concentration gradient, what else is it going down? Positive, negative. It's attracted to that negative, right? It has an electroattraction, so it's going to rush in. But as it rushes in and become positive, then we're going to have a little conflict, right? Positive will start basically repelling. So it has to come to some sort of equilibrium. Same thing with potassium. And actually, that one I show a picture of. Okay, let's just look at this picture. Kind of a nice picture. The idea is, if you open a potassium channel, we already said it's going to go out, and basically down its concentration gradient. But if you look at it, potassium is positively charged. It's also sort of attracted to the negative charge inside. And so, at the beginning, there's so much potassium it's going out, but there reaches a point where an equilibrium, where, okay, we're losing because of the concentration going down, but positive's attracted to negative, and so it sort of stays, it comes to some sort of equilibrium. So, with that said, you don't need to know, okay, back to this, you're not going to need to know for this class the so-called Nernst equation, which actually, if you know the numbers of how much is in and out of these ions, you can actually use it to predict what the actual voltage is. That's what it's used for. I'm going to use it to illustrate basically point number 2, and I'll tell you what that is here. So, look at this right here. What if, this basically says, what if we only had sodium? What would the inside of the cell be if we allow it to come to equilibrium? Remember, it's going to go in, positive attracted negative, until this becomes so positive that it repels it. And if you look at this, you basically allow it to go in, and it will come to equilibrium for just sodium, the inside of the cell would be plus 62. Okay, if we just had sodium. If we just have potassium, it's going to go out, come to equilibrium, the inside of the cell would be minus 90. So what's the real answer? What is this closer to? It's minus 70. Which one of these is it closer to?
62.
Is it closer to this one or this one? Minus 70.
Oh,
Minus 90. And what this is indicating is, and this is what we're going to put down, there is more potassium leaking out than sodium leaking in. And that's really creating where it's more negative. Okay? Because remember, sodium leaking in, equilibrium would be pretty positive, but the potassium leaking out would be really negative. Really, you can see it's closer to this. So instead of being minus 90, some sodium comes in, but not enough, and so it's still only minus 70. So this is why it's a complicated point. I'm going to put it in words and see if this starts to make sense. So the sodium-potassium pump sets this up. But the reason really it's minus 70 is that the cell, these are the leak channels, the cell is, I'm going to use the term, more permeable. And, in a way, let's put leakier, for a word that we can understand, to potassium compared to sodium. So, in even sort of simpler terms, more potassium leaks out compared to sodium leaking in. And again, lots of complications, and if you really read the book, they talk a lot about Nernst equation and all that stuff. That's really, when you look at this and what I want you to know, when I'm asking you what are the two factors that create this resting membrane potential of minus 70, are these two things. The sodium-potassium pump, pumping 3 sodium out and 2 potassium in, basically is creating part of this electrical, making it more negative. But really the idea is that now the leak channel exists, more positive potassium is leaking out compared to positive sodium coming in, and so the result is that the resting membrane potential is minus 70.
So point 2 would be the leak channels.
Yeah, well, the leak channels and using
Yeah.
the leak channels, that more potassium is leaking out than
Right.
sodium is leaking in.
But if you're gonna simplify that definition, it would be leak channels.
Well, but you can't just say leak channels
Yeah.
without saying, but you could put it, leak channels with this statement here.
Right.
More potassium is leaking out than sodium is leaking in. Yes, ma'am.
When you're talking about the leak channels, are you talking about the things that activate them to allow it to, or just that it gets through?
No, see, we're going to talk about, actually gated channels in which we control. These channels, these leak channels, just exist. So they're just there. They're literally like a hole that allow these ions to go in. And it's a small hole, so not everything goes back down to 0, right? And the pump's working. But it is sort of a natural hole that allows these ions to cross.
And is that the nodes of whatever?
No.
So we're talking about something completely
We're just talking, we're just talking about in general, the whole nerve itself. Before anything happens, we have leak channels, and this includes, in the dendrites and cell body and all the way through. Okay? And again, even if you don't quite, I mean, it's a little abstract here, just have that down and we're sort of done with that and we can go back and revisit that. But those are the two things that create the resting memory potential. So, what we're going to talk about is we're going to fire a nerve. When it comes down to it, I'm going to redraw this and again get the basic circle of life, and that's what we're going to do next. And so, when we're talking about the nervous system, skeletal muscle, cardiac muscle, smooth muscle, it's going to be these four channels. These four ions that cross are going to create all of that: muscle contraction, nerve contraction, everything else. That's why we had to do a little chemistry in that first section, so you get the idea of what ions are, these salts, these electrolytes, why we need them, and how we're going to control them crossing the membranes, the nerve or muscle. These four channels. And so, you have this. I want to erase this. I want to make this back to where we were, nice and simple. So again, when you're practicing this and kind of getting it in your head, this is the basic you want. You want them to remember we have negative charged proteins and we have what ion?
Potassium.
Potassium. Okay? Outside the cell, and again, that means higher concentration. We have three ions which are high outside the cell, which are?
Sodium.
And chloride. And now we're going to be talking about, we already talked the lead channel. Everything now is going to be the idea of control channels, which we're going to talk about beta channels, in which something's going to trigger them opening, and then the ions are going to go down their concentration gradient. And that's what we're going to talk about when we're actually firing a nerve. Actually, not anything leaking, but we're going to open channels and we're going to close channels. And it's just these four. That's all we're going to be talking about for all the, the next half of the course as far as function.
What about -70?
And oh yeah, we can put negatives. For right now, for a typical neuron, if you do all this up, it adds up to -70.
Okay, but
Later-
it's not always going to be -70.
Exactly. And so we'll see in -90 and all that. It's the same principle, but different cells. When we talk about neurons, else are like the skeletal muscle, they'll have a different value there.
And then you'll show us how to get the value.
You're not even going to have to quantify it. You'll just see, because again, that's going to be a level that's like another course to use the Nernst equation. So
Okay.
you're not going to have to to particularly know it. And so what we're going to see will be electrical signaling, will be ions flowing across. Now we have to get some terminology here, and the terminology sort of is here. Okay, kind of these terms here. But I'm going to sort of close this up a second and use the terminology that we're going to figure out here. And so what I'm going to do is basically, I'm going to make a scale we're going to be using a lot. And basically this is going to be, on the y-axis, the membrane potential. What is the inside versus the out? So the membrane potential. This is going to be something we do. This will be time. And let's put 0. Let's put -70. Let's put +30. And just, as a thing. And we'll put right now -55 for fun. Okay? Now, the resting membrane potential of a neuron is?
7.
-70. It's down here. Okay? Now, if it were 0, you have nothing, no potential. But we are -70, and that means because it's not equal, we call this state, it's polarized. It's more negative, so it's polarized. So, let's say we see a deflection, we do something, and we're going to talk about what we could do, that it basically gets more positive. The inside of the cell gets more positive. Okay, what we're going to call that is depolarization. And I could put this up just for that. Okay, just so you're spelling it. When you're getting more positive, this is depolarization. So I'm going to put deep, but you can write depolarization. And I'm going to write depolarization over here as well. Okay? Now, if it becomes, and we'll show you how, let's say it becomes more negative back to where we were. That's called repolarization. The final term, and then we're going to go back to the circle of life, the final term is if we do something, which we'll show, we're opening and closing channels. If we open channels and we're actually more negative than we started, it's actually called the cell is now hyperpolarized. It's more negative than it even was at rest. It's hyperpolarized. I'm going to put that right up here also. Because what we're going to do here is practice using the circle of life. Because
It's hyperpolarized if it's below negative 70.
yeah, for this one, and in general, if it's
Yeah.
below whatever the resting memory potential is. And you can kind of see that it's below minus 70, because this is resting. So, to figure out basically what causes this, depolarization, repolarization, hyperpolarization, it is going to be, and right now we'll even make it, let's kind of get this separated. It's going to be opening channels. And these are the only 4 channels we have. And you don't have to memorize what these are on a separate note card, because you can look at that and tell me, okay? For depolarization, what channel would we open?
We would open the NAC channel.
If we open the sodium channel, sodium comes in, makes it more positive, that's going to be a depolarizing stimulus. So we can put it, open sodium channels. And again, I'm going to write open sodium channels. And just reminding us, we're looking at that. Why? Because sodium will go in. And that makes it more positive. We're going to see opening sodium channels, boom, it's going to depolarize the cell. What's the second channel we can open that would do something similar?
Chloride. Oh no. Potassium.
Okay? How many people say calcium? If you open calcium, which way is it going to go? Does that make it more positive? Yes. Okay? Positive going in means the cell is going to be more positive. So this coming in or this coming in is going to be two ways that we can depolarize the cell, make it more positive, and that means going up. Okay? Just like sodium came in, that's positive. Same thing. Calcium's high out here. So we could just make a mess. We could, I'm going to put this in a different color. We could open calcium channels. Okay? Calcium goes in. That's the second way we can depolarize the cell. And we're going to see some places have sodium channels, some places have calcium, some places have both. Okay? When we're going to be talking about an action potential here, a neuron, I just want to just basically give you a hint. We're only going to be using sodium channels. Okay? But theoretically, and as we go through describing more of it, both of these are depolarizing. Other places we're going to use that as well. But you can see that. You're not memorizing that list because you look up here and you can tell. When we think of the idea of in fact, if I draw it this way, if we think of the idea, again, this is repolarization and this is hyper, the bottom line is we're making the cell, the inside of the cell, more negative. What are the 2 channels that we can open to make it more negative? Potassium and chloride. If we open up chloride, chloride goes in, and that makes it negative. If we open potassium, potassium goes out, that makes it more negative. We're losing positive. Okay? So we're writing those. And that's it, by the way. Those are the 4 channels for anything we're doing. We're basically opening these one or more of these channels. So again, to make it repolarized or hyperpolarized, we can open potassium channels and keep writing the ion flow because I want to make sure you get it. Potassium goes out. Or we can open chloride channels, chloride goes in. As far as right now today, and we're talking about the action potential, what's going on through the axon here, we're only going to use potassium for that. So literally we're going to see, before class is over, when we look at, this will be actually what an action potential looks like. This will be what's happening while that's transmitting all of this, and we're literally going to revisit it and talk about that sodium channels are going to open, sodium comes in, potassium's going to open, potassium goes out, and that's, again, that's going to be a nerve firing. Just those two ions, just those two channels that are opening and allowing these ions to flow down the concentration gradient. Sodium being first because of the timing of this, potassium next. And we're literally going to see, like dominoes, these ions go flowing down the membrane. Very, very cool. Okay? Does everyone sort of get the terminology and also the idea of where, you're going to have a lot of information in this chapter or throughout the course. You don't really want, like, I've got to remember a separate note card on this, really, and then maybe I'm going to mix it up and confuse it like hyper-hypo. If you know the circle of life, you can reason it out. As long as you know depolarizing is more positive, you're like, okay, well, there's only two ways. Sodium's going to go in or calcium, and so I know that's the thing. Same thing on the hyperpolarization or repolarization. More negative, it has to be this and this. And so starting with here, you can already figure this out. So don't, you can do whatever you want. But I wouldn't have like a stack of note cards for this class, really. Just know how to reason it from that. Any questions right now on this? Is this making more sense? You see how this, it's going to start to flow a little more like physiology and less overall. By the way, guys, you guys right here, you have to stop talking while I'm talking, okay? Because it's, again, I can sort of talk over you, but some people back there might actually just be disturbed. So you really, and I'm big on that this semester, and I got like 400 students. So it is a problem, okay? So you have to try to limit your conversations. Again, not as much for me, although it is kind of rude, but also for the people around you, do it. You can talk to me and shout out.
Sorry.
Yeah, you can talk to me.
Sorry, I didn't know what you were saying.
Go ahead.
So if something is hyperpolarized, to bring it back up to -70 or whatever you're starting is, it's still called depolarization then, right? Or is that not something you have to deal with?
You won't have to use that term, but it will be that way. You'll be depolarizing it from there to
Okay, I
get
just
up
wanted
to
to make
that.
sure that I wasn't thinking repolarization was always back at the start. That's only if it's higher then.
No, I mean it's basically. We just won't use the term that much, but yeah, it will. Anything that's making more positive is a depolarizing
Okay.
stimulus, even from here.
So that little itty
That little
bit
bit would be depolarization
Okay,
back up to
that's-
resting. Yeah.
Alright.
So that's in general. It's just that for hyperpolarization, it is sort of the idea of where it's below normal. And so all those are the words. And I just did all that with us.
Below normal,
So,
below normal negative, does that make sense? Because
More-
or are you not gonna always start with negative inside?
No, you are.
Okay.
Yeah, it's below the resting membrane potential of the cell. Okay, which in more cases right now will be -70.
So an action potential will include depolarization, repolarization, and hyperpolarization.
Yes. And so this will be, and we'll actually look at this by the end and actually just do like 1, 2, 3, 4, 5 steps. That's literally what it will be. It will literally look like this. And that's
Over and over again.
over and over again. And we're going to see either we're going to have it or nothing. And if we have it, they're all going to look the same. And it's not going to lose strength. That's what we're going to get to next. That's going to be the key of an action potential. But all together, because we're going to talk about other types of potential, we're only involving these 4 channels. And you see which way things are going to go because it makes sense to us. So we move on. And the next thing I want to just mention, and then I need to erase this, and we're going to basically talk about how we get from here to here. And then we're done for the day. So before we do that, we do have to introduce kind of what I started to, and what you started to talk about too, as far as controlling. You talk about all these ion channels with these 4 ions. There are leak channels, as I've already described, that are always open. Okay? It doesn't allow everything to go. I mean, they're specific for sodium or potassium or calcium specific, but the idea is we're not controlling it. They exist in that membrane. But the next 3 are going to be things we call gated. And that means when we look at these channels, okay, if you look through the membrane at these channels, there is a gate. It's going to be closed until something triggers it to open. And there are 3 types of gated channels. One of these is mechanical. Okay, this is actually some physical pressure to it, to whatever, this is on, whatever neuron. And if it does that, some physical pressure, it's going to open the channel, and, we'll just keep sodium there. And sodium can flow through it, or whatever it is. When it comes down to it, these are going to be, when we do the sensory system, that's how most of those operate. If you're hearing my voice now, in your inner ear, there are these hair cells with fluid around it that's vibrating and basically bends them, and boom, basically those will open. So we'll see in chapter 10 a lot of these mechanically gated.
So the leak channels are the 4 ion channels in the membrane?
Well, not all membrane has all 4. The leak channel, in other words, all these are only the 4 ions we're talking about, but when we're talking in general about leak channels, they are not controlled. They're always in the membrane, allowing some things to just flow down the concentration gradient. But not all neurons have the same amount of all 4 leak channels, etc. So they vary in what they have. But the definition of leak channel means if they exist in that neuron, we're simply going to allow, if we have a calcium leak, we're going to allow calcium to go down its concentration. So far, we've only talked about sodium, potassium, but again, they're just there. We're not controlling them. Mechanically gated, we're going to control. How are we going to activate it? Physical pressure. Okay. Again, it could be our somatosensory we'll do in lab of just touching, or we're going to actually stick some pins in us and stuff like that. That's going to trigger them. The last 2 types: chemically gated, voltage gated. Chemically gated means we're going to have some of that neurotransmitter, like serotonin, that's going to bind to the receptor. Boom. That's going to trigger open. If we look kind of over here, that's what we're going to see next week when we actually get to this side of the coin. Once those neurotransmitters are released, basically, they're going to be binding to these channels, opening them, and allowing ions to flow through. So these are chemically gated. Again, they're going to be talked about next week when we actually, a little tonight in drugs, when we actually talk about neurotransmitters working. The last type of gated is what we're talking about today. These are voltage gated. They're going to respond to change in membrane potential. And what that means, a lot of times in the book, when we see pictures, you're going to see like plus, minus. Just to indicate that it's voltage gated. And so the idea will be, the inside of the membrane, these are embedded, remember, is like minus 70 for these neurons. Okay? Well, if we change that potential to a certain level, it's going to trigger it open. And so let's say we've got some positive coming in, and right at the gate, it's now only minus 55. Something like around there. Guess what might happen? Gate opens, sodium rushes in. So it's controlled by the membrane potential. So if we get this membrane potential, but at the axon a little higher, minus 55 or even more, boom, we're going to trigger the channels to open, and then we're going to actually see some stuff happening. If we don't, okay, if we're at minus 70 or minus 60 or something that doesn't reach that number, the gate's closed. So that's what we're talking about voltage gated. Is the change in membrane potential, which again is the difference inside versus out. If it changes to a certain level, which is for these particular receptors, whatever they are, these channels, then it will open up. If not, they stay closed. That's how we're going to control the opening and closing of these channels that are going through the axon.
So what you're talking about, is that the same as leak channels?
No, because leak channels don't have a gate. Leak channels are like in the membrane, like let's say we have a leak channel, it's just open all the time. And so a little sodium can always flow through, or
a
little potassium. There's no gate, it just is always going on.
So then the gated one allows more sodium?
It will. So it allows nothing, and then it will allow a rush of sodium.
Oh, okay.
Because this is a tiny, it's almost like think of a tiny leak versus opening up something in a dam and allowing the water to rush through like a waterfall. Okay? And that's why, that's the idea that, and remember how this is going to be quick too, right? We're talking thousandths of a second. When this triggers, boom, this is going to happen all the way down and, within, again, thousandths of a second. So it's going to be very quick because we do have this waterfall of ions.
Does it have to be more negative, lower than negative 70 to pass through? Like
No, it has to be, and we're going to see this as we go through, it's got to be minus 55 or higher.
Oh,
So we need to get a depolarizing by at least plus 15, right? 70 minus 55. So plus 15 or higher, and we're going to kind of go through that for this to fire at the, the action potential here. But I'm ahead of it a little bit because we want to actually get to all the way. Yes?
so like does the leakage just keep it at a certain rating so it
The leak channels reach some sort of equilibrium. It helps keep it at a certain level. So I guess in a way you're like over pumping, and then it kind of comes back to an equilibrium over it. Just kind of like how the resting potential did, right? We pump so much, but then it settles to an equilibrium using those leak channels.
And then if something happens that requires more sodium to go in, then the gates would open?
Yes.
Depending on if it's voltage or mechanical.
Or chemical. And then we're going to have to eventually reset it. The sodium potassium pump is always working in the background. Okay, 24/7. 30% of our energy that we eat is used to drive the sodium potassium pump in every one of our cells to create these gradients. In this case, we're going to use it to actually do electricity. Okay, we're going to do neuron. And so I got to clean this up. Clean this up. Clean up this figure. I'm going to keep the circle of life. I'm going to keep this here right now. And actually, as I keep it there, I'm actually going to label 5 things of it. And we're going to do that last. That's going to be what's happening in an action potential, and that's going to be after we have to get to it first. But we're going to describe that 1, 2, 3, 4, and 5. But first, when we talk about electrical signals, change of potential, there are 2 types. There are make my cell better. There are graded potentials and action potentials. 1, 2, 3. There are 3 main differences that I have listed, and I'm going to illustrate them, actually just putting them up here and explain them. So graded potentials. Well, first of all, why I put them there are graded potentials happen only in the dendrites and cell body. So I'm just rewriting that, but I'm just doing it where it is. And of course, action potentials only occur in the axon. Okay. Now let's get the other 2 points, because that's going to be critical. We're starting off from graded potential, and let's explain this. So they're of variable strength and size, and we can write this. Let me just write these again and explain this. Variable strength, and as it's traveling, because it's going to be traveling this direction, it's going to lose strength. So let me show you that. So first of all, what we're doing here to create these graded potentials are opening up ion channels. Okay, something's going to trigger one or more of these ion channels to open, and we're going to have a potential, basically, and we'll kind of indicate that by, again, kind of an up-down. Okay, depolarization, repolarization. That's going to be opening up channels, and one of those ions or more are going to flow in. Okay, now the idea of this is how big of a spike is going to depend upon the signal strength. Let's say we have a weak signal versus a strong signal. Okay? So let's say we have a whisper versus the kidney. Okay? Okay, now we're just doing hearing, but it could be anything else. The idea is that the strength of the graded potential, is going to be directly proportional to the stimulus strength. because it's, the stronger it is, the more of these channels it's going to open, and the bigger the change is going to be. Should make this a little better. So that's what we mean by variable strength. It's dependent. The variable strength is proportional or related to the signal strength. What's causing these channels to open? The stronger the signal, the greater the potential that we've created, because more ions more channels open, more ions went in. Okay, everyone have that. That's the first point. Okay, why we mean by variable strength. Now, the other key factor is, okay, let's look at these guys. Let's get rid of the nucleus. We know what it is. We know we're traveling here. Here's the axon. And there's actually a particular part, the very beginning of the axon is going to be the trigger zone. And we're going that direction. Okay, the cell body's adding these up. Now, the second bullet point is, as this travels in that direction, it loses strength. Okay, even this one, it loses strength. Okay, and that's going to be an important part. Okay, so it's variable strength, but it's losing strength as it travels. And the two reasons why are sort of given here. One of them is that overall, when you look at the membranes of the dendrites and cell body, they are leakier. Okay, they have more of these leak channels. Things start leaking the other direction. So ions leak one direction or the other to make this, but it's going to sort of counteract itself a tiny bit as it travels. And so, again, it's leaking, a little leakage, and so you're having a smaller amount by the time we get to the trigger zone. Okay, the other one is a little abstract to me, but it's called cytoplasmic resistance, so that as this is traveling through the cytoplasm, and obviously there is actually resistance to it, it dissipates some of the energy. Okay, now again, I think if you're really good at physics and electricity, you have that idea of the resistance and its relationship and blah blah blah. I don't really care. The idea is that's the second reason. I guess I should care, but I mean I don't care that you really understand the physics of it. Just that when you ask the two reasons, or if you're trying to figure out the two reasons of why it loses strength, again it has this leakage and this resistance, which by the time it gets here means it's not as strong. Okay, that's the three about the greater potential. If you look at action potentials, okay, the opposite. Action potentials, we already said they occur in the axon. It's going to be all or nothing. If they exist, they're going to be the same strength, the same size and shape. And unlike the others, they do not lose strength as they travel. So the key will be, if by the time, and we're going to show it visually on this, by the time you reach the trigger zone, if you have reached the threshold voltage of minus 55, we are going to have action potential that looks like that. Okay? And it's going to look like that. And by the time we get to the end of the axon, it's going to look exactly like that as well. So it's not losing strength because there are really no leak channels, or very few along the axon to allow leakage to go. And the other idea will be, and that is kind of what I put here, we'll kind of show it, that for greater potential, we're opening up a few channels, okay, depending upon the strength. Okay, when you look at the channels that are along the axon, there are many. They're lined with these channels. And really, and again, I kind of want to put a picture ahead, what it's going to be like. Okay, and then we'll come back to it, are going to be like dominoes. I should just put two copies of that picture. So it's not like we have one big action potential. The idea will be, it will be like dominoes, that once we trigger, we hit threshold kind of at this first region, sodium comes in, and it's going to then activate the next set of receptors. And then we'll have another one. Then that positive coming in will activate the next, and so it's like dominoes falling because of all of these channels. Once one goes, they all go. And there's enough channels that that's why they're the same strength. The trigger causes the exact same amount of sodium in and potassium out all the way down. And that's sort of the idea of that. Same size and shape and does not lose strength. It's either going to happen and looked at or not. Okay? You know, if the signal is too weak and we don't reach threshold, we're not going to have like a tiny little action potential. Or if it's really strong, we're not going to have an extra big one. It's all or nothing. If we do not reach threshold, nothing. If we reach threshold, the same. Let me show the picture that illustrates this. And then we do the 5 steps of the action potential, and then we're done. And then before Monday, you're going to do everything we did 50 times. So that when we do this at the beginning Monday, we're going to start from the beginning. 2 things of resting potential. And we're talking about going through it. You're going to do it with me, and then we're ready for the next amount of information coming in. Okay? That's going to be the key. So whiteboard it. So let's look at this. Let's take a look. It's sort of how they describe it. So, how does the nerve fire? Some sort of stimulus are going to open up channels, okay, causing in the dendrite cell body, causing greater potential. Okay, if this reaches threshold, by the time we hit the beginning of the axon, the trigger zone or axon hillock it's called, you're going to have it. Otherwise, not. So let's show it. So they show 2 examples here. So in this case, there was some sort of signal, and when they put a probe up here, it actually was above threshold, okay, when they were kind of doing it. Actually all the way up to -40. The dot, dot, dot is the threshold voltage. But we're not at the trigger zone, are we? It's traveling. It's losing strength. By the time they put a probe here, it was barely at the threshold, but that's not where we need to be. At the trigger zone, it's below threshold. That means no action potential. We're not going to transmit this at all. But what if it was a stronger stimulus? Look at it up here. It's losing strength. Okay, the key is, by the time it reaches the trigger zone, it's -55 or higher, boom, we're going to have dominoes. We're going to have this action potential travel within milliseconds to the end. So it's all or nothing. Okay? It's either if you do not reach threshold, nothing. If you reach threshold, boom, action potential.
So, then what is the middle where it barely reaches? Like what happens with that?
But nothing's going to happen because
So it goes yes, very strong action potential, nothing and nothing.
no. Because look where the probes are. They're saying if they're measuring the membrane potential like at here, that was enough. But the key will be it doesn't matter what the membrane potential is here. Only matters by the time it reaches the trigger zone, okay, which is this, because it's losing strength. So at the trigger zone, if you haven't reached -55, no action potential. It doesn't matter what was up there, okay, because the axon's not up there. It starts out pretty good, but because of those 2 reasons, losing strength, by the time you reach this level, that's all we care about for threshold. That's where those voltage-gated channels are. If we do not reach it, which we don't, no action potential. If you had a stronger stimulus, again, we don't care about what's happening up there. We only care what is it right here. And in this case, it was above -55, and you'll have an action potential.
Oh, I was just gonna ask that if it does go through the trigger zone, like it does make it, does that mean that it will send that, I don't know, go to the other cell right to create the synapse? Like, would that be going towards that? Like,
Well, what do you mean? I mean, if it doesn't reach threshold, that's the end.
yeah.
There's
But
no
if it
signal.
does, is
Then
that
it goes-
to the synapse?
It will go, and we'll finish it. Actually, we'll do it next time. We'll talk about it. We'll go to the synapse, and we'll talk about a releasing neurotransmitter and affecting that cell. That's sort of the second half of that. And so, and again, nerves are made to either it will work or it won't. Yeah.
Under action number 2, when it says does not lose strength, that's during or after the trigger zone, not before, right?
Right, because these are greater potentials. So right before the trigger zone are the greater potentials that have those characteristics of variable strength and losing, variable size and losing strength. Once you get to the axon, that's how it has these 2. So yeah, because of all these channels, it's not going to lose strength at all, and again, it's going to be the same size. It's going to look like this. Okay? This is what the action potential is going to look like. I don't care if we had a strong signal that was way above threshold or we barely made it. Doesn't matter at this point. they're all going to look like that. They're all going to have the same amount, amplitude, and the same shape. And if you look at the book that has 9 steps or the figure, that's describing it, that's nice, but I don't care, I don't want to do it that way, exactly, because you know how I am. I only care that we understand these 5 steps. Okay? 1, 2, 3, 4, 5. And that's what we're going to write down. As I take a drink. And that will be what that's going to be where we stop today, because that actually makes it to the end. And then we're going to review it all and then take us to the other side. And that's going to be chapter 8. Okay? Nice flowing stuff. Everyone sort of gets it. It's just kind of getting some factoids. So let's actually do this again. And we've already kind of done it. We're going to use the circle of life here. And just remember, we're only going to use, when we're looking at these channels, there are not any calcium or chloride channels in here. All of it's just going to involve the sodium and potassium. Okay, I'm not even going to write that. So, again, now we go this. We've reached threshold. Okay, and we have this. What do we call this? Depolarization. And what's causing the depolarization? As far as what channels are opening? Sodium channels open, and sodium goes. Okay, I care about ion flow that you get it. That's what's the trigger. Okay, we're going to have voltage-gated sodium channels. If we reach that voltage, boom. Sodium rushes in, the cell becomes more positive. That's depolarization. Okay, number 2. Well, we stop. So what do you think happens to those sodium channels?
They close.
That's all it? Sodium channels close. We'll make it a little more complicated next time, but that's really what happens. Sodium channels close. So that's going to end that depolarization. What's number 3? Repolarization. We'll get to that. Repolarization. And what channel is going to open for that? That's simple, guys. You get it. Potassium channels open, and potassium goes out. Boom. We're repolarized. Of course, we do a little bit of what in number 4? Hyperpolarized, so hyperpolarization. It's getting more negative. And remember, I said it's only potassium, so I guess potassium channels stay open a little bit longer. And so more positive leaks out. So now we're actually below threshold. So all of this are potassium channels opening and sodium going out. Hyperpolarization, potassium continues to go out, or something like that, so you get it. Continues to exit cell. So, only one more thing to put on this, but overall, that's the idea. We have, they'll talk about that's membrane potential and blah, 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 but I just care about this. We have depolarization, sodium's going in, sodium channels closed, potassium channels open, they go out, potassium goes out, making it more negative, repolarization, continues to go out, and makes it hyperpolarized, okay? Now, of course, I don't even put it sometimes, potassium channels closed, okay? But now look what's happening. We're slowly getting back to resting membrane potential. So we're slowly becoming more positive. So who's involved? Sodium. Sodium leaks in, okay? So actually, we got it right. That's what I mean. We get the flow. It happens, that's why it's kind of slow. Kind of at that voltage of minus 90 or something. What's that positive sodium say? I'm going to be sucked in through that leak channel a little bit more because I've got that attraction here. Up to resting, and now we're at equilibrium. So the book doesn't describe that in the 9 steps, but if you read the paragraph below, and I know you all do, it actually talks about that idea of coming back to resting membrane potential. So sodium leaks in until we're back to resting. And those, again, we're going to revisit this and add a couple complications, but not much more. Those are sort of the 5 steps of the action potential that basically allow us to travel with 1,000ths of a second without losing strength. Okay? 1, 2, 3, 4, 5. All involving sodium potassium ions. Okay? We haven't even dealt with all the other ions, which we'll deal with later. But as for action potentials, that's sort of all we care about.
Would you call, so the last step 5, would you call that return back to normal?
Yeah, it's returning back to resting memory potential.
So that would be the repolarization.
Repolarization, well, again, we weren't really we're gonna term it, but, it's gonna it actually depolarizes back up the rest of it. But all you have to do is to go, is that's how we get back up the rest of it. Let's go to
I know how that is, that's how it was Monday.
So, again, the test scores will be up by the weekend. You can see the test next week.